So yeah, finally. So hi everybody. I am Zhangzhi Fred. Call me Fred. Currently a PhD student at Duke. Today I'm really excited to describe a kind of a new protein language model (PLM) that capture the PTMs. So the story begins with PLM. I guess most of the people here probably heard this word a million times. So protein language model is a gigantic deep neural networks that's been trained on millions of protein sequences and be scaled up to millions and billions of parameters. So there's good representation of the proteins, the interactions, and the evolutions. It's pretty good. It's been applied to all kinds of applications. Something we care about. ESM4 predict the protein structure, and PLM also be applied to predict the mutation of the proteins. And there's a question we really care about in the lab is, can we, there are some type of cancer-driving proteins that are pretty disordered. Can we design binders to those proteins such that we can solve the cancers, right? And there's another, there's a paper from the lab is called PEPLM. So we basically fine-tune the protein language model on the target binder pairs. So the model has been trained to uncover the binder given the target sequences. So it's a good application of protein language model for protein design. And the problem is all those protein language models, ESM2, protein GPT, all of them are only encode the 20 types of 20 types of amino acids. So we know like probably biology 101, there's PTMs, post-translation modification. You may a little bit backgrounds. So PTM refers to a type of chemical modifications on the protein backbones, and it kind of alters the protein structure, localizations, and also functions. And also if you look at the POXO3, POXO1, this is the proteins that's being phosphorylated, which drives the, enhance the protein stability and also activities. So we really need a model that understands the context, which in this context is PTMs, such that we can enable predictions, design on top of the PTM, the modified proteins. So yeah, that's what I'm gonna present today, PTM-Mamba. So first of all, let's talk about how we represent it. So I'm a computer scientist. I'm dumb. So the way I represent PTMs is don't look at the chemical graph. The way we think about it is just a token. We know like this PTMs is installed on this position, so I replace it with the corresponding PTM notation, and then we go to UniProt and we search for all the possible PTMs, and we do a little bit of screening. And at the end of the day, we got 80,000-ish sequences, PTM sequences. And if you look at the figure on the right, it's a little bit long tail distributed. So a couple of PTMs that's been like kind of dominating this database, which kind of makes sense because that's how the scientists collect the data. So yeah, we got the data. So let's cook the model. So recently, there's a type of state space model, kind of pretty popular input in generally machine learning. It's been really good in modeling the sequences. So in this work, we are using the new state space model Mamba. Due to the time constraint, I will not be able to talk too much about the architecture, but the simple idea is Mamba is a type of a recurring model that understands the sequence from the left to right. And we use the Mamba here in this context, but we want the model to understand the sequence from both ways. So what we do is that we take 2 copies of the Mamba and we feed into the sequence into one and flip the sequence and feed it into the other one. And we're combining them to have a bidirectional Mamba block here. And also, since PTM-Mamba understands both the wild type amino acid and also the PTMs, we don't want the model to kind of bias towards PTMs so that we leverage the embeddings from the ESM2 for the regular amino acids and focus and kind of force the model to bias towards the PTM tokens. So here we figure out a way of combining the embeddings from ESM2 and also the PTM-Mamba. So we get the embeddings out, and we calculate the weight that we basically using the embeddings to first compute the weight for assigning to like the PTM embeddings and also the ESM2 embeddings, and we dynamically combining them using what I call here is a gated embedding fusion module. So yeah, I scale up the model to like 200 million parameters and train on the existing PTM sequences. And now we have a model that understands PTMs. But we do want to know if this model understands the PTMs. So first of all, we look at the embeddings. So those are the proteins with both the wild type representation and also the PTM representation here. We fit into the model. And so we get the embeddings and project it into 2D space. And we see that they are spatially pretty close to each other, but not the same point. Meaning the model kind of understands the nuanced differences caused by the PTMs. And also we look at the PTM tokens, and we visualize into 2D. And you can see on the bottom right, those are the wild type amino acids. Those are the model didn't learn much about it. That's why the embedding is pretty narrowed down to the subspace. But you look at the PTM tokens, the model learns pretty diverse representation. And if you look at the, I can't point it out here, but like the phosphorylserine and the serine, those are the two PTMs of the same class, and they are spatially close to each other. We didn't force the model to have the kind of knowledge, but the model kind of figured it out during the training. So we think the model kind of learns the embeddings of the PTMs, and also we want to evaluate the model, and we want to see what kind of utility of this model. So basically, we train the model, fine-tune the embeddings from PTM-Mamba on kind of a PTM side prediction task. Here we present the phosphorylation prediction, and we compare with the ESM2. Without a doubt, it's going to be better. And we also did the experiment on acetylation side prediction, and it's still better. And we want to see some other more interesting practical application. So we fine-tune the PTM-Mamba on a task called disease association prediction. So given a sequence, you want to predict whether or not this protein is associated with a disease. So this binary classification, we also compare with ESM2 and also the simple one-hot representation, and still we outperform the ESM2. And this is the other one, druggability prediction, given a sequence, whether or not this protein is druggable. So also still we outperform the PTM, we outperform the ESM2. So it's gonna be a little quick. I speak a little bit fast, but the take-home message here is, if you wanted to understand so for example, if you have a bunch of proteins and you also have the PTM knowledge about it, you probably want to use PTM-Mamba because it gives you more context about the PTM and what's the consequences, what's the effects of the PTM on the sequences. So that's the take-home message. So with that, I want to thank to all the members in the lab and also my PI who took me to the Duke basketball game. So and also our founder. Thank you, guys.
Thank you. Time for questions.
Can you comment on where you think the PTM information in UniProt is coming from? I'm worried that most of it's going to be from bottom-up mass spec, so you actually lose all the dependencies between PTMs within, all the crosstalk that people are interested in is not actually reflected in that data. It's sort of artificially sticking multiple PTMs into a single protein based on separate peptide measurements.
Yeah, you mean, can you say that again? Can you summarize your question?
Yeah, so I think the measurements that you're using from UniProt are probably coming from peptide level measurements.
Yeah.
Right? And so they don't reflect dependencies between PTMs in a protein. Is that a problem?
Yeah, exactly. I think when I collect the data, the PTMs are pretty independent. So probably got 3 or 4 PTMs on the sequence, and they are independently represented in the UniProt. So that is the question, and I think I guess we can't do much about it because this is how the data get collected.